Hello everyone, my name is Nejc Stopnisek and I'm postdoctoral associate at the Michigan State University, working with Ashley Shade in the Department of Microbiology and Molecular Genetics. So thank you for giving me the opportunity to present to you our plant microbiome work, in particular how we go about to study the common bee microbiome. I hope I will convince you at the end of my talk about three things. First, the common bean is an important crop, an appealing model system for studying plant microbe interactions. Secondly, that plant rhizosphere is a very diverse and complex system. And third, and finally, that plants consistently recruit a number of microbes from the soil, suggesting an intimate relationship between the plant and these microbes. So as mentioned before, I'm a postdoctoral researcher in the group of Ashley Shade, and what our main research team is microbial stability and what role plays microbial diversity in this process. So if you look at these figures here on your screen, after an ecosystem goes in perturbation or stress, the community in this ecosystem goes into a period of fluctuation. And this fluctuation can then lead into two end results. First result can be, and the desired result is a stable state, which means that the functionality of the system it's very similar to the pre-perturbation state. However, on the other hand, the community can go into an alternative state, which is unstable and usually has negative consequences for the system. So not just for the system, this has effect also on the plant health. So what we want to understand is how microbial communities respond to stress and use this knowledge then to manage these communities to be stable and resilient in the future. So our approach to study the plant microbiome resilience consists of three parts. So first of all, we want to understand how these communities look like. So who is there present? Are plants selecting for some taxa? Are the host genotype effects on those community assemblies and community compositions? Does seasonal effects or even, or as I said before, plant growth affect these communities? So we are approaching this with using the amplicon sequencing. But what we do next is exposing the systems to stresses. And why do we do that is to understand more how the system works and how the functionality of such microbial systems look like under stress conditions. So what we are doing here is using the amplicon sequencing combining with the metagenomics. Finally, we are very interested in the functionality of these systems and trying to better understand what microbial functions are involved in this stress conditions for the plants, and which can be also having effect or implementation into plant health. So we are using metatranscriptomics and metabolomics for this part to investigate. So as microbiologists, we are very fortunate to be surrounded by experts in plant sciences. We are part of two large research efforts. One is the Plant Resilience Institute that is funded by the Michigan State University, and the other one is the Great Lakes Bioenergy Research Center, which is funded by the DOE. Our research model system includes a variety of plants that are including the bioenergy crops such as the switchgrass and miscanthus. We are also studying apples, but as my title of the talk already suggests, we are focusing lately very much into common bean or the physiological vulgaris. So what do we care about and why would we focus about the common bean? So there are two reasons. Firstly, common bean has importance for the society, and it has economical, agricultural, environmental, but also health benefits. And secondly, it is an attractive system for science due to its unique diversification and cultivation history. It is a model to study the impact of plant evolution and domestication of plant-host microbe interactions. So diversification of this plant started around 160,000 years ago, when the common bean ancestor started migrating from its origin in central Mexico through Central and South America. Interestingly, two domestication events happened simultaneously, approximately 8,000 years ago, one in Mesoamerican region and one in the Andean region, which then furthermore actually isolated this germplasm and speciation. Here in the figures, you can see the nowadays germplasm that exists in the wild, and you can already see the distinct lineages between the Mesoamerican and Andean. And this separation is not just in their genotype, but also in their phenotype and traits. So additionally, there is a major problem concerned regarding the common bean, namely that the yields and production area are predicted to be decreased due to the impact of the global change. That being said, it's abiotic and because of biotic stresses. So, as mentioned, many stress related to climate change are affecting the production of the common bean, as you can see also here. So, however, there is still germplasm that, which has resilient traits and can be bred into the modern cultivars. This can be achieved through traditional breeding. And you can see that in this picture where some of the beans, let's say in the drought conditions, are experiencing stress and then dying off. However, on the other hand, we have some germplasm that is tolerant to the drought, and this trait can be then bred into the modern cultivars. This is a tedious strategy and usually takes decades to achieve. The other alternative and approach, what we believe it's also very advantageous with many other groups, is to do that with microbiome engineering. And what do we think about, or what do we consider as a microbiome engineering? There is a nice paper by Wallenstein in 2017 with a very nice figure. So what we are thinking, again, with this engineering is we can use different stimulations, either in live microbes, microbial communities, or even substances that will boost the environment functionality and diversity. And this will then become, again, healthy and natural looking, as it's in the picture of A. We know as well that our modern intense agriculture systems tend to be degraded. That means that they're depleted in the diversity and in their functional redundancy. So any stress that will hit such systems there's a high likelihood that these functions can be lost forever, and that will even more degrade the system. So engineering is a process where we can actually boost the system or even improve the degraded systems into a more natural, healthy environment. So in our research, what we are focusing is to understanding the natural occurring microbiota for these purposes that has also, of course, benefits under stress conditions. So, however, there are some problems with this. And this is because the rhizosphere and the soil ecosystems are one of the most diverse ecosystems on the planet. In this figure, you can see the diversity of microbial diversity across different ecosystems. And highlighted in the arrows are one, it's the rhizosphere, and the other one is the soil. And you can see clearly that those ecosystems are truly very diverse. And this diversity, then, how do we find the ones that are actually essential for the plant? So what we think is by defining the core. And what we mean with this is to find a set of microbes that are found in most samples of a particular plant, and will help to identify them, the plant-associated microbes that should be prioritized for the future research. What is an additional aspect of the core is that it reflects underlying functional relationship with the host, and so we can use them really clearly for that purpose. The common bean, though, the common bean microbiome, though, it's very under-investigated. And so we don't know clearly who is associated with this important plant. So to shed some light on this, we started a project where we are looking at actually the common bean microbiome and defining the core. So we use different approaches. We combine different approaches, and one of them is biogeography. Biogeography is a study of species distribution across geographical locations and was traditionally used in the animal and for plants. However, also bacteria or microbes can show some biogeographical patterns. Here in this map, you can see a distribution of marine environment associated microbes, some that are more distributed across different oceans and some that are more localized. Next, what we are also using and combining here is the using of microecological concepts and mutual modeling, plus co-occurrence network analysis. Both of concepts I will introduce to you in a minute. So how does our experimental design look like? We focused on we selected two very distinct genotypes, as I said, one from the Mesoamerican and one for Andean diversity panel. The Mesoamerican diversity panel is comprised from the Eclipse genotypes, the black bean, and the CELRK, or California early light red kidney, which is a red kidney bean. They're not just genotypically very different, but also phenotypically, and their traits are very different. So if we find that there is that the plant genotype has an impact on the communities, we should clearly detect that with these two genotypes, only two genotypes. So we grew those plants at 5 different locations across US. And these locations are also considered as one of the top producers of the common bean. This location not just different in their geographical distribution, but also in their soil properties, from pH to different phosphorus levels, organic matter, but also their climate and management. Some obtaining fertilization, some not. Some do have irrigation and some not. So our approach now was to harvest those beans, isolate the DNA from the rhizosphere, and amplify the 16S rRNA gene for understanding the communities of bacteria and archaea, as well as ITS region to target the fungal community in the systems. We use these amplicons to sequence on the Illumina MySeq and analyze those by clustering the reads into operational taxonomic units, or OTUs, at the 97% similarity. What an OTU, or operational taxonomic unit, is, it could be equivalent to a species in the animal world or plant world. So this data set has been used then for various analyses, which I'm going to present to you right now. So the results of this study are as following. So what we found is that the study showed that there is a strong effect of growing location on the bean rhizosphere communities, but there is no effect of the host genotype. We have used meta-analysis, which includes samples from the US, our study, plus a study done in Colombia, and we have found 48 taxa that represent a cross-continental core. Moreover, these core taxa are not hub taxa and mainly are low abundant. Results from the combined co-occurrence analysis and neutral modeling then also suggest that these core taxa are in intimate relationship with the host plant, and rather than being key species in the soil environment. So let me just show you now some data supporting these results. So to investigate the community composition and the effect of the soil properties, the management or the climate, we have used ordination analysis and Permanova. We were surprised actually to see that the common bean genotype did not correlate with the community composition as it was found in recent studies, but the studies differ as well in their growing conditions. So as seen in the figure here, represented in the principal coordinate analysis, and how we read this figure or the graph is like every dot represents a community structure. If the dots, the samples are closely together, that means that the community composition of these sites are very similar. More distant the points are, the more distinct the communities are. So by color coding, you see that points with the same color tend to be organized or grouped together, suggesting that they're very similar by location. So that makes us first believe that the core might not be present. However, despite significant differences in microbial composition between these growing locations, the community composed of a very similar microbial taxa, and thus we were still then speculated that we can still maybe find a core, since you can see here in the Venn diagram that almost more than 2,000 taxa have been found across these sites. So to determine the core and to investigate the core, we relied on the occupancy abundance relationship. And an example of it is presented here in this graph below. For each taxon or the species or microbe in this community, mean relative abundance is calculated and then plotted against its detection frequency, which we term it as an occupancy. So the higher the points are in this graph, the more prevalent the taxa are in this data set. So this relationship is also a starting point to understand the community assembly mechanisms by using the neutral modeling, which is represented in the line. So we have used the slow and neutral model, which was also implemented by others, especially Burns et al., which assumes that for taxa with high abundance, we should expect also high occupancy, and then also vice versa. So taxa, there are always some taxa that don't follow this rule, and those deviate from this. We can call them as deviation from this neutral model. And those taxa are in particular interest for us because these are assembled through deterministic processes or selection, and with that have ecological importance. So when we apply this relationship to our data, what we first see is that for approximately 22,000 OTUs or species in the system detected across 30 samples, many taxa are shared in white, and only few of them are strictly genotype specific, as highlighted in the colors orange or black. This just confirms the previous data with the ordination analysis. So moreover, when we apply the neutral model, the community overall fits pretty nicely to it. However, there's still almost 14% of taxa that deviated from this model, and most of them, interestingly, are predicted to be above the prediction, or the prediction where selection for these microbes can be assigned to. So due to the large number of taxa that we find in this system, we decide to use a very, very strict threshold, and this is for defining the core. And this is all the taxa or species that are having occupancy of 1, meaning that they are found in every single sample that we have. And you can imagine we have collected the samples across very different geographical locations with very different properties and from two very different plants. So everything what we found here is very strictly. Still, we identified almost 250 bacterial and archaeal taxa that fall in these very strict thresholds. And even more, 171 of these taxa are predicted above the neutral model. So next, what we wanted to do is to investigate the importance of these core taxa. So recent research actually suggests that microbial hub taxa are important drivers of plant health. And how to determine if they are hub taxa? Well, it's usually done by correlation network analysis or co-occurrence network analysis. So for those less familiar with the network terminology, a hub is a species that is in the community highly correlated with many different members. And it has an importance and implication for community assembly, for community functionality, but also overall then for our ecosystem. So in many cases, biological networks tend to be modular. And a module, it's a group of taxa, species, nodes that are highly correlated between each other. And here in this figure depicted in red, highlighted in red or blue. There are also species termed as connectors, and those are the species that are connecting two hubs or two modules, excuse me. And everything else that is there are termed as peripheral taxa. So there are two parameters that can be used to distinguish these four categories of nodes. This is the within, the ZI, and between, the PI module connectivity, as seen here in the scatter plot. The result is this plot, the scatter plot, which is split into four sections and each representing these four categories. So the outer layers being the most important, either being the module hubs, the network hubs, or the connectors, and internal ones, that is the peripherals, which tend to be low connected within the module, but also between the modules. So when we use this model, this method on our data, we found that our core taxa are not hub taxa, except we found one taxon out of 270 that belong to the family Chytinofagaceae, which was determined as a model hub, and then one connector species belonging to an Enterobacter family. Out of 270 taxa, actually, 75 only showed statistically significant correlation with other taxa, and most of them have not been included in those. So suggesting that predominantly this core taxa, which tend to be very ubiquitously and found with the common bean plant, are peripheral or not very well connected to the community. What are the peripheral taxa in the ecological perspective? These are the specialists. And so can we think of that these are specialists for the plant? Still the question that we are trying to address. So to validate our results, actually what we looked for studies that use as well next generation sequencing approaches, using the common bean, of course, and rhizosphere. We found actually a perfect study that was ideal to compare with, and it included the 16S RNA regions, same to ours. It has additional 7 bean genotypes, which were sampled at flowering time, as well as what we did. And but the difference is that they were grown in the controlled environment in Colombia. So results from that actually surprised us. And since we found a large overlap in the membership between the study, despite being so different, we found 48 taxa that are shared between the cores of each data set, having the Colombian core and the US core represented by the taxa with the occupancy of 1. And who are these taxa? Predominantly, these taxa belong to the Proteobacteria, but there are still like nuances in their relative abundances between the data set. Some, like Microbial down below, tend to be more abundant in the US than in the Colombian soils. However, Sphingomonadales, members belonging to the Sphingomonadales, tend to be more abundant in the Colombian samples versus compared to the US. So what is the reasoning behind it is not clear, but we could speculate that these taxa are in intimate relationship with the host, and that the host, depending on its needs, then modulates their abundances and activities. So with that, we kind of represent and presented a manageable number of taxa, which our future research can actually target on. So, but we are not the only one in search of the core, of the plant core. Many research studies have done similar research in other crops and plant species, and all with the same actual objective, to find a set of taxa that could have application potentials. So what we have done next is like we use a publicly available data set and ask if there is actually a universal core rhizosphere microbiome. So to do that, as I said, we searched for public available data and found 18 different studies, including 18 different plant species that were grown in 9 different countries, represented in total by 240 samples across 12 studies. What we did, we processed all the data using different workflows from MuSearch, Mother, Chime, and classified the reads by using Silvo database. The results are actually pretty surprising to us. So the thresholds that we have used are slightly different to previously. Here, our threshold was that the occupancy of a taxon must be greater than 0.5 within the study. And so half of the data should have this taxon within the study. Plus, the other threshold should be that if they have this occupancy, they should also associate with all the plants in the metadata that we collected. So looking at this data set, we see now highlighted in the black that we found 83 taxa that are shared regardless of the plant that we included, and that's been 18 different plants, being either wild plants or domesticated plants. Even more, when we looked at when we separated the plants into, let's say, domesticated or the crops and the wild plants, this core membership drastically increased and inflated. So when we look at the now domesticated plants, more than 600 taxa have been found using this threshold. And on contrary, the wild plants have lower, but still high amounts of membership that can be termed as a core with 200 taxa. So this was really surprising to us, and we are currently investigating that more into depth. But who are those cosmopolitan and dominant or the dominant taxa? So they are predominantly belonging to the alpha perturbativa group, still tending that we see differences from plant to plant. Some plants have more alpha perturbativa than others, but what is the consequences of these differences is still unclear. Mentioning already at the beginning, we're very interested in functions of the core microbes, and also in particular now in this data set. And how to understand that from the data that we have collected right now from amplicon sequences, that's difficult or almost impossible. So what's needed is to do further studies. And what we initiated, it's a study that involves, again, common bean, and was conducted in the summer 2018, where we were looking at how microbial community actually develops on the plant throughout the plant growth or development, where we also included the previously bean genotypes, the CELRK and CLIPS, and grew them at the 2 exactly same locations in the Michigan, which is the Saginaw Valley and Montcalm. This time we did not just focus on the rhizosphere, so the root architecture part, but also looked at the phyllosphere or the above ground communities, as well as the endosphere communities or the communities that reside within the plant. So we are here questioning whether and how the communities develop on the plant through the plant development. Does that has consequences for the functionality of these community compositions and of these community structures, but also what are the potential of these microbes in the systems. So for now, we have only preliminary data from the amplicon sequencing, and what is nice that these data support previously results that the plant genotype had no effect, and that the site has a strong effect on the community composition. Moreover, what we see that the compartment where we sampled and analyzed, as well as time, they also have very strong community structure effect. So how this community structure differences transition into functionality is still unclear, and this is hopefully with obtaining the metagenomics data and metatranscriptomics data, we will be able to address that. So to summarize now my talk, it's what we have found is under fill conditions, plant genotype does not influence the common bean rhizosphere community. We also find that the plant core taxa have potentially plant beneficial traits for which they are then recruited by the plants. That's been supported by their high occupancy, the predictability or deviation from the neutral model. Even more, what we can also argue is that the plant core taxa are not important players in the soil microbial communities because they are weakly correlated with other taxa in the network analysis. So what we have found as well is that the plant development and root compartments are strong drivers of the community composition in the rhizosphere environment, but we still don't know how that affects the functionality of it. We also found with the meta-analysis that there are different plant species that recruit similar microbial taxa. Whether there is actually existing a plant cosmopolitan taxa, it's yet to be determined. So from this descriptive, let's say, studies, we actually now initiated a number of very excited projects looking at more of functionality of these core microbiomes or microbiota. And we are currently working on assessing the aspect of seed microbiome and it's impacting the abiotic stress tolerance in plants. We are trying to understand how actually plants communicate the needs and recruit soil beneficial microbiota, in particular looking at the role of phytohormones. We are also trying to understand how actually much microbes contribute to observed tolerance in plants. And moreover, what we want to actually achieve as well is sustainable growth of plants, right? And this is by using native microbiome members and using those for sustainable growth. So with that, I'd like to thank you for your attention. Thanks to Ashley Shade for giving me the opportunity to work on this exciting project, all the members of the Shade Lab, as well as the funding agencies for supporting our work, and for, of course, the resources at Michigan State University. At the end, I'd like to invite you to visit our GitHub repository if you are interested in any analysis, data wrangling, and the bioinformatics pipelines and workflows that we use in the lab. With that, thank you very much. If you have any further questions, I will be available on my email. Thank you.